hopefully one for a lot of you, as we have a lot of new sequencing toys out there in the market now. And we have a really great lineup of scientists to tell you all about what it has taken to get these instruments in their groups. Before we get started, we are going to have our sponsor give a talk. And Watchmaker is our sponsor for tonight, this afternoon. Please hold the questions till later, and I'm hoping we have time for like a nice discussion afterwards. Thank you.
Hi, I'm Jen Pavlika. I'm a senior product manager with Watchmaker Genomics, a genomics tools company headquartered in Boulder, Colorado, with over 100 employees worldwide. There we go. Our focus is on engineering enzymes and tailoring solutions to address gaps and needs across a variety of challenging applications, with a specific focus on improving data quality. Now, as I'm sure you're all aware, core facilities and sequencing service providers have a unique set of needs. One centers around performance, namely maintaining data quality when your samples are coming from a variety of sources and spanning a broad range of input amounts and template qualities. Another centers around price, namely the continuous need to reduce costs that come in various forms, including maximizing sequencing economy and reducing costs associated with both reagents and operations. And last is workflow. So workflows that have low turnaround times, low hands-on times, are easy to implement and easy to automate, are hallmarks of workflows that are going to ultimately save labs on their operational costs. Just as important are workflows that come with expert technical support to further ease implementation as well as reduce downtime. Now, many of these needs are actually in opposition with one another a lot of the time, and many solutions can deliver two of these or address two of these needs, but rarely all three. And Watchmaker is working to change that. Our first focus area was in DNA library prep, where users are typically limited to workflows that are either scalable or deliver excellent sequence accuracy. Sonication is regarded as the gold standard for unbiased genomic DNA fragmentation, but it is difficult to scale and requires costly equipment and consumables. Enzymatic fragmentation is an attractive alternative, which addresses the process bottlenecks as well as the monetary barriers to adoption, but it has its own inherent challenges, namely in the form of the formation of sequence artifacts. Our goal during development was to harness the workflow benefits of enzymatic fragmentation and deliver data quality on par with sonication. So any workflow to be considered easy to use must be both tunable and consistent. And to the left, we're showing the tunability of our solution to generate library-sized distributions of a wide range using only fragmentation time and temperature. And to the right, we're showing consistency across a wide range of DNA inputs from 100 picograms up to 500 nanograms, demonstrating our robust performance across what is essentially a 5,000-fold DNA mass input range. And as I mentioned before, many of the existing enzymatic solutions suffer from higher prevalence of sequence artifacts. One such artifacts are chimeric reads that can be present at up to 10-fold the rate in enzymatic fragmentation libraries relative to sonication control libraries. The presence of these artifacts at these inflated rates can hamper your ability to disentangle what are true chimeric reads indicative of variants from those that are just artifactually generated during library prep. And this can also impact your sequencing economy. With an eye on this during development, we were able to minimize the formation of excessive chimeric reads and deliver rates on par with sonication and much lower relative to other enzymatic fragmentation solutions. Now, another source of sequence error that's not specific to enzymatic fragmentation but is relevant for DNA library prep comes in the form of PCR-induced errors. We leverage our Equinox library amplification solution across our full NGS portfolio for high-efficiency, low-bias amplification. And here we're showing base substitution rates relative to Kappa Hi-Fi, stratified by specific base substitution events. And what we see is a reduction in misincorporation rates by up to 40%, and this is mainly driven by a significant reduction in C to T base substitutions that is particularly relevant for oncology applications. These improvements and reductions in sequence artifacts and in overall sequence accuracy imparts improvements in variant calling accuracy. And in this independent study conducted by the Broad, comparing our enzymatic fragmentation solution to sonication, specifically for PCR-free whole genome sequencing library, what they observed were SNP and indel F1 scores that were indistinguishable between our enzymatic fragmentation solution and sonication, indicating sequence accuracy on par with the gold standard. Next, we focused on RNA library prep, whose workflows are typically onerous and time-intensive, often taking multiple days to execute. We wanted to improve on these workflow bottlenecks while minimally maintaining, but ideally improving upon data quality, specifically with challenging samples such as low inputs and FFPE. Because of our desired focus with FFPE samples, we focused on streamlining total RNA sequencing workflows that leveraged combined rRNA and globin reduction process. And we streamlined this workflow specifically by combining enzymatic steps, as well as reducing enzymatic incubation times and reducing the overall number of bead purification steps in the workflow. This is nice from a user perspective. It also helps to minimize non-specific loss throughout the workflow. And these workflow improvements resulted in an overall solution that can reduce your turnaround times by 2 to 3 hours. It can reduce your hands-on time by up to an hour, and actually reduce your pipette tip savings by 1,000 per 96-well plate. It's also worth noting that we focused a lot on FFPE performance during development, and all of this work culminated in the implementation of a novel FFPE treatment step geared towards reversing residual crosslinks in your samples to make more RNA available to support library preparation. Bless you. In addition to workflow improvements, we also see data quality improvements. So looking at a high-quality, whole blood-derived total RNA sample with inputs ranging from 500 nanograms down to 1 picogram across a variety of solutions, we focus first on wasted bases. These are bases that are characterized by either passing filter but not aligning to your reference, or bases that are aligning to your rRNA and globin reference, neither of which are relevant for your research applications. As you'll see, as you reduce that input amount to 1 nanogram, the number of wasted bases is inflated across all of the chemistries, but this rate of increase is inherently smaller with the watchmaker solution relative to other solutions. This translates to improvements in unique gene detection sensitivity, especially at lower input amounts where we're delivering up to 3,400 additional unique genes with 1 nanogram inputs. When we look across the whole cohort, all input ranges assessed, all workflows, and we perform a hierarchical clustering analysis, a few trends pop out. One is that our 1 nanogram libraries cluster with 10 nanogram libraries prepared by other solutions, indicating that you're getting similar information with 10-fold less RNA going into library preparation. The other trend is that 1 nanogram libraries prepared by other solutions cluster together, but not with the rest of the sample cohort. This indicates that you can prepare a library with 1 nanogram inputs and sequence them, but that the data that you're getting isn't truly representative of your original biological sample. And the third trend that we see is that the watchmaker solution is the only solution wherein 10 nanogram through 500 nanogram samples cluster together, meaning you're getting similar information out across a 50-fold range of inputs. So if we come back to this idea of these 3 main driving needs for core facilities, Watchmaker has addressed them across both the DNA and RNA NGS workflows that we've implemented, delivering improved sequence accuracy as well as improved gene detection sensitivity, with simple, scalable, and automatable workflows, and at a reduced cost, both in terms of the operational costs associated with implementing these solutions, and reduced reagent costs as a function of the fact that we manufacture all of our own enzymes in-house. We have many more solutions that I didn't have time to cover today, and we're particularly excited about our mRNA library prep chemistry launching this summer. Our recently launched DNA library prep solution for cfDNA samples or for sonication prepared libraries, and our high purity RNase inhibitor, particularly suited for single cell RNA sequencing applications, launching this fall. So please come by booth 404 for more information, as well as to inquire about our early access programs. Thank you.
Yes? Okay, so what am I, so we're not doing this now?
I need a Zoom link for Breakout, for the third speaker.
Can you? Yeah, I think so. We need to. Thank you. Okay. Hello from me as well. First of all, absolutely thrilled to be here today to this celebration of core facilities and shared resources. And I wanted to thank the organizers for putting this together, and also Anoja for the kind invitation and setting up this exciting talk. So I will discuss tonight about how the G4 ended up in our facility and how it's working. And I think it will make a good panel because the colleagues here have also very interesting systems to share. So these are our disclosures. We work with a lot of good people out there. We paid for the system, so we didn't get it free to say something really nice tonight. Or we can accept other systems or more of it. So yeah, we're very open. This is the algorithm that we followed to decide on a system. Of course, this sounds a little bit dry, but it's just very briefly, where does this system fit? Not only your core facility, but where your core facility fits in the whole ecosystem, and how will that system, affect it? What is your primary need? If you can do it like with single sentence, I need this to support my clients to do X, or I want to increase my productivity there, or I would like to capture the libraries that do not fit on my NovaSeq. It's very important to set your primary goal, and we'll discuss this later on, and then you can make it into specs. You evaluate how it fits on your workflow and how it affects your direct reports and the other people working there. And of course, is this your only sequencer? Do you have other capabilities? We will discuss this a little bit more. Do you want to see data before you buy something? Do you want to speak to people, or do you want to see this in your system and work on your ecosystem and work with it? And the most important things for me are, how do you define success? You need to know this beforehand. It's not, okay, I've been working with a system for a year and I see a drop in my cost and I see increase in this. I think if you're doing a postmortem, it doesn't always end well. It's very important to say, my number one priority is to increase my productivity there, or to capture that, so that enables you to make specific metrics, evaluate every quarter, and if you need to make any corrective actions, you can. So I tried to make this into simple questions that might be useful. I'm happy to share the deck. So where will the sequencer fit as part of a larger core institution puzzle? Can you clearly define what the sequencer will cover? Can you render these needs into specs? I think that is quite important. After you phrase it, you have to break it down. What does it mean? What types of libraries do I have? And I want to run on this system. How often? What is the volume? Of course, you would like to run perfectly everything and optimize everything, but if you prioritize it, I think it's easier to keep track. How important is the number of samples per month? How important is your turnaround time? What is the problem you're trying to solve? What times, wait times expected to pull per run? I think this is very important. Can you wait to pull? Do you need to run like right here, right now? How much can you wait to pull, and how big those pulls can be, and how big are the libraries you're usually pulling, and so forth. Cost projections, your business plan, a cost per sample, personnel needed, purchase cost, service cost. So you take virtually the sequencer you're interested in, you place it virtually in your mind, in your ecosystem, and then you're trying to understand how will that affect things. How will it cost, and what changes I need to make in my workflow? Oh, this system needs pooling, or this system needs, as we'll discuss, conversion. How will my timeline change? How will my workflow change in order to accommodate for that? What if the system needs maintenance or upgrades? This is very common for every new system. If you get a first generation sequencer like just coming out, there might be firmware updates, there might be system updates. If there are 2-3 days downtime for that, how can you accommodate, and how will you accommodate? We discussed about those, and what will success for purchase mean to you, and how will you be evaluating the success for the system? So the ecosystem, I think, is very important. This is our ecosystem. We're a spatial core facility, and this is what we do. We do single cell spatial. There is training, there are different things, but I think the key part that we're going to discuss today is access to technology. Usually, clients just drop tissue samples on our bench, frozen, fresh, or FFP, which we then dissociate, and we do single cell or single nuclei from fresh, frozen, and FFP as well. We try to automate as much as possible. And then for spatial work, again, we cut, stain, do our own tissue microarrays, and we're trying to automate again as much as possible. And there are multiple modalities, which those two are the main drivers, along with the single cell, that we wanted to internalize with the sequencer. So our primary need was we want the sequencer to cover these assays, but most importantly, our protocol optimization. Because clients just drop tissue, and we do single nuclei protocols, for instance, or we do Visium protocols, or we wanted to have the ability to run a sample today or two, and tomorrow know whether it was a good run, and then we can scale. So if we write this, we want to internalize NGS to enable rapid protocol testing and optimization, as well as the completion of time-sensitive projects. The PI coming like, oh my God, I need data yesterday, and what can we do about this? So the system we wanted, if we make it into specs, we wanted to be very cost-effective with very small runs, like 2 sample single-cell runs. That was the most important thing for us, and it doesn't mean that it's important for you. So that's why I think the ecosystem is key in this discussion. We want very short turnaround of time, because if we know the protocol is good, we can work, or if we need to pilot 2-3 times, we don't want to waste 2 weeks. Support larger runs if required. Doesn't have to, but if we can also do production on the same system, same cost, or better, that will be great. And in terms of quality, these assays are expensive, as you know, single cell and spatial, so we cannot reduce the quality for our clients. The data should be equally good or even better if possible. Support and then beta test. So this is the timeline, how things played out for us. We identified we need to internalize sequencing. End of 2020, 2021, we were the first beta test location for the G4. So we got to actually know the system. When we had money a few months later, about 6 months later, we reevaluated every sequencer out there. And based on our beta testing experience, we thought the G4 would be good for us. We purchased it with our own funds. It was deployed around September 2022, and we started working on it, reaching maximum throughput around January. So after that, the system was run multiple times a week. We just upgraded, so this is important for you to know, our system, because we got the first G4 leaving the factory, it was not the same as the one you're buying today. So our system passed through, it was an earlier version, it looks a little bit different as well. So we just got upgraded to the same that is on the market a few weeks ago, which is the commercial system. And we're looking forward to the max reads. This information, I have a star, you can find it online. I don't think it's important. It is very important why we chose it, but the most important for us is how the samples fit in the sequencer. So the sequencer has 4 flow cells, independent, and each flow cell has 4 lanes. Our assays, which are usually 10x single-cell Visium, and the geomics is a little bit more complicated to discuss about fit, so I'll stick with single-cell Visium. If you're using the F2 flow cell, it's 1 sample per flow cell. So if you have 1 sample, you use 1 flow cell. If you have 3, you use 3. The cost is fixed per flow cell. The prices are MSRP, you probably will have pricing based on volume discounts. I'm using what's on the website. And if you use the F3, which is a new flow cell, it's 2 samples per flow cell. So if you want to do 2, or you can do 4, or you can do 6. For us, that was key because we usually run 1 or 2 samples to pilot, and then we usually run 4 or 8 when we're scaling. So this number was the golden number for us. Other systems, they have like multiples of 6, or we had to wait to pool. So our pilot run, we would need to have more samples in order to wait to pilot. And because this was mostly selected to be very fast, we were also sold on the very fast turnaround time. So 15 hours means that if your library prep, we end at 5 p.m., 8 a.m. in the morning, you have results. So you can plan your next day based on how that previous run came to be. And max reads, we don't have access to it yet. Max reads will give about 3 billion reads on 4 flow cells, which means 16 samples. So 16 samples, again, is a good number for us because this is the maximum throughput we have in a day in the lab. In our experience, first of all, our setting might be very different to yours. We run a very small number of different library preps. A lot of those, but we just run 10x, Visium, and single cell, maybe TCRs. Usually that's 80% of the sequencing volume. We got the first system that was ever commercially sold, which means that it's worse than what you're getting, and I don't know what your experience will be. In terms of, now we matched, so we know, but we have it for 2-3 weeks now. In terms of primary needs, it works. From 1 to 8 samples, it's great, which is usually our daily throughput. So you finish your day, you run the sequencer, morning you have data. It's about 46 hours. Instrument time, data quality, everybody has been quite happy, our bioinformaticians and the other bioinformaticians. Library conversion, I think if you're using it as much as we do, I think it's a process worth automating, and we're focusing on that. It's not that it's very time consuming, but if you want to run the sequencer every day, somebody has to be doing this every day. And I think a robot might be better than our personnel. Now I think, it's always, should I wait for a new system? Should I wait for it a little bit? It has its plus and its minuses being an early adopter of something. First of all, we had the ability to work for a tailored purchase. If you buy early, you discuss your goals and what you want to achieve, and you get potentially good advice on that. However, as I mentioned, we passed 2 or 3 different upgrades, and each upgrade needs time. Or, for instance, if we notice something and we have a request, that request can make it to the commercial system and your system, but this means you might need downtime for that request to get into your system. Also, some of the key things you like, the F3 that we wanted, the F3 just became available. So there is a gradual rollout of your wish list as well. Another good thing is there are a lot of eyes on your system. Are you happy? Are you not unhappy? How does it working for you? Do you like the new menu? If you don't like the new menu, how would you like it to be? We like that interaction. So it has its pluses, it has its minuses, but for us, I think it's a success. And now we get a lot of the question, would you go for the new generation desktop sequencer? There are many good options on the market at the moment. The main answer is these new desktop sequencers, Element has a strong offering as well, are very disruptive in terms of pricing, in terms of workflow, in a good way. what options you have if you have a small core or a medium core where your volume, cannot fit a very large competitive system. I think the main key take-home message for me is I don't think a solution fits every core. If you do this exercise, I think you can match it to your needs. Super thanks to our team and of course to the whole STU and the Precision Neural Medicine Core, which is our more extended ecosystem that we belong to, and looking forward to the discussion with you later. And we're hiring, so if anybody's interested, let me know. Thank you.
No.
Yeah.
That's Delty? Perhaps you want to go first then?
He's first.
No,
He's
those-
before her.
You can use the laptop.
Where are we connecting? Perhaps she can go first then.
What do you need?
We need to connect the laptop.
Okay, good. Okay, good. I hope it's working now. Oh, good. So I'm Lutz Frönicke from the DNA Technologies Core at UC Davis. It's yeah, good. Helpful. At UC Davis, and we're part of the Genome Center. The Genome Center has a variety, of course, proteomics, metabolomics, bioinformatics, and bioinformatics is, of course, our most important partner because they analyze a lot of the data that we are generating. And we have been historically at the forefront of new technologies, and as part of that, we also acquired the element BioAviti. So we're running a SQL 2, we're running a Review since a few weeks, we're running nanopore sequencing, BioNano, and we're doing all types of high throughput library preparations. So we need to able to run a vast variety of sequencing libraries. And regarding short-read sequencing, we're generating about 10,000 short-read sequencing libraries a year. And we are running these on 3 MySeqs, a NextSeq 550, the Aviti since about half a year. And we never upgraded from the HiSeq 4000 to the NovaSeq because that thing, we never liked it enough, but we're sequencing. It was a horrible design in my eyes, but that's personal. But we're shipping fully compiled lanes for the NovaSeq S4 to other UC cores. And we're doing a lot of that. Last year, we got involved with COVID sequencing for the California Department of Health. And we told these that, we would love to help you. This is a special part of the COVID sequencing project where they wanted fast turnaround times. to, for example, monitor the borders in the airports, the samples that come in. And we told them we need a new sequencer, and they luckily agreed to support us in the purchase of one sequencer. That's how we got the Element Bio RVT. And we're doing the CDPH sequencing for that, but we're also running customer libraries and our own libraries on that. And what, as you may have heard, the reagent prices for the Element Bio are very, Element Bio RVT are very appealing. So we are running a single one of these sequencers, so we're paying the standard rate, the base rate, and we're offering the sequencing of a flow cell with about 1 billion read pairs for $2,000 a run, and with 150 cycle kit, which you would use, for example, for 10x genomics libraries for $1,400 a run. That includes the conversion of the library, which is the circularization and everything else. And the main advantage, the prices are, in this case, are very similar than sequencing on the NovaSeq S4 on the biggest NovaSeq flow cell, but for our purposes, we can do much faster turnaround time. That's how the reagents arrive. You kind of have unpacking parties. And what you see at the small, basically the cartridge is packed there. That's the cartridge has a considerable size. We fit about 30 of these in one standard lab freezer. And what's different from Illumina sequencing that most people will be familiar with, you need to circularize the library. And that applies to both Element Bio libraries at the moment, as well as Illumina libraries. And we so far have actually only sequenced Illumina libraries. So you need this additional circularization because the clustering protocol involves rolling circle amplification. And this is one of the many differences from Element Bio sequencing to Illumina sequencing. And I'm not going to go into the details. The company will be able to tell you much better about their unique and fascinating chemistry. The input requirements are a little bit higher compared to Illumina sequencing, but since you're getting 1 billion reads out of there, you're pooling multiple samples anyhow, and it's not really a problem to achieve these concentrations. So you're starting out the circularizations with 15 microliters, best case scenario at a 60 nanomolar concentration. That takes about 75 minutes, 25 minutes hands-on time, and the good thing is we have not run into any surprises. We've run more than 70 flow cells. It's a rock-solid protocol. Whenever we started it, we got good libraries out of in the end. Afterwards, you're doing a qPCR for quantification, or since a few weeks, Element Bio is also supporting a protocol where you can skip the qPCR and do single-stranded DNA qubit protocol. That is faster, takes only about 20 minutes, but at the moment we're still evaluating it. We're doing both in parallel. And that is also suggested so that you get to know your libraries and verify that the protocol works as expected. The qPCR is supposedly, I've never done it myself, but it's actually simpler than the kappa quant protocol that we usually use for Illumina. You're working with the Element Bio software. The sequencer graphical user interface is straightforward, and it guides you graphically through the loading process. What you don't have at the moment is an equivalent of the SAV system, where you can monitor the run in greater details during run or after the run. What you have is the Element Bio Cloud here at the bottom. That allows you to monitor the run and gives you Q30 curves and phi-x error rates and other run metrics in real time. You can access it from your home remotely. And then there's a software basis to FastQ, which allows you the conversion of the raw data into FastQ files and demultiplexing. That is very straightforward. If you're familiar with BCL to FastQ, you will find the syntax of your commands very familiar, and it's very fast. It's wonderfully fast. And this is Cyranush, a member of our lab who is running the sequel most of the time. starting the sequencer. You see it's guided. You can't really go wrong. And this is what you see during the run. You see Q30 curves or phi-x error rates curves and expected yields. And this is the same what you then see from the BioCloud remotely, the same information. The FastQ files look a little bit different than the Illumina because the quality scores are higher. As you can see, so the majority of the bases will get an M, which is 44 thread score, quality score, and that is obviously indicating higher quality, which is great, but very, very few old or badly programmed pipelines might run into problems because they don't expect these high quality scores and they will balk. But for example, 10x Genomics very quickly put out an update for their pipeline, and it's now perfectly happy with these data. So we did run more than 70 flow cells by now. As mentioned, we're running actually exclusively Illumina libraries so far because there was so far not a big advantage to run the Element Bio libraries. There will be very soon because soon the circularization for the Element Bio libraries, you will be able to do this on the system and the run without the extra step. What we are sequencing most frequently are, of course, the COVID sequencing protocols. We're doing a lot of 3' and RNA-seq. That works fantastically. We're running this only on the AVT anymore because it's an extremely cost savings. Lots of the 10x Genomics libraries are sequenced on their GBS RAD-seq. This is usually something more difficult to sequence because there are low complexity stretches in there, as well as TN-seq. And we have not done, some stringent testing. For example, on the HiSeq 4000, the TN-seq libraries required 40% of phi-x spike in. We have done one run on the AVITI with only 15% phi-x. The data were perfectly fine, so potentially we could go down further, but we have not really examined it. Quality scores are great. So on our runs so far, we have Q30 rates of 92 to 98%, and Q40 rates from 75% to 89%. Mean quality scores range from 40 to 43, so above what you get from in Illumina, because they don't even go that high. And barcode assignment rates obviously depend on your barcoding reagents, but this is everything fine. These are our yields that we got, and the scale here goes from 500 million reads to 1,300 million reads. Green line is 1 billion reads. As you can see, at the beginning, we were one of the very first customers. We had some issues, and these were really resolved. It turned out that we had these problems. A few labs running such a sequencer had these problems. And Element Bio started a big research, more complex study on it and figured out what the problem was in the end. And it turned out to be that we did, like several other labs, dry the beads a little bit too much after the circularization reaction. You're precipitating single-stranded DNA, and apparently that can cause artifacts if it gets too dry. Since we're avoiding that, the yields are beautiful. We did not any extensive validation of the sequencing data because, first of all, there are others published already and people can do this much better. But of course, we had to verify that our COVID sequencing works just fine before we going all in with AVITI sequencing. And in this case, we use the same libraries, sequence them on the AVITI on the NextSeq 500. And what you see in blue here, for the same library, and this is very mixed quality samples. So what we're getting from CDPH is, partly horrible samples that are actually from antigen tests. We're sequencing the leftovers from antigen tests. Nothing dedicated collected for sequencing. We're sequencing in part very crappy samples. Anyhow, for the RVT, we could assign then the data of variance to variance for 89 out of 94 samples, and for the next SIG we could only do this for 79 out of 94. That indicated it works great for us, but I don't want to say in this case that the sequencing data are dramatically better on the RVT. I can't say this. What it turns out to be, you can see the median coverage based on the same number of reads. We have here 1000x coverage on the RVT and 648 coverage on the next SIG 500. It is that this pipeline, this standard pipeline for the data analysis, viral recon NF core, does a very stringent quality trimming. So it trimmed just a ton of the next SIG data away. They were no longer available for analysis, and if you run this pipeline, you're definitely better off with RVT data, but can't generalize that. Yeah, that's the pipeline. Yes, so we did not do any further sequencing comparisons other than our customers have verified that the 10x genomics stuff works just fine for them. You learn more from difficult samples. For example, we just recently, for Casarin, sequenced a sample where we did not have enough library. So we loaded only about half what's recommended, and we still got 600 million clusters out of there. High quality, I hope so. And if you're sequencing libraries that are longer than recommended, the sequencer had absolutely zero problems with it. Yes, so far with the sequencer, we had not a single low quality run with quality problems. Quality was never a problem. As you've seen at the beginning, we had in the beginning some low yields runs, and that was due to the bead drying problem. Very, very likely in almost all cases, but sequencing quality was never ever a problem.
Thank you.
Yeah.
So our next speaker, Michael Quell, is actually connecting from UK, and he'll be on Zoom here soon. Give us a minute.
Hello, my name is Bud. Okay, I think that should be about right. Can you let me know if it's not? My name is Bud, and I'd like to tell you a little bit about the Open Trust.
for our National Health Service in the UK. Our sequencing lab is equipped with 14 NovaSeq. NovaSeq X is coming on Monday, 6 MySeq, 2 HiSeq 2500s, and 1 HiSeq 4000. We also run PacBio Revio, so 1 Revio so far, 2 more on the way, and 12 Sequel 2E. And in addition, we have ProMeFi and GridION. So a little bit about me. I work as principal scientist for sequencing R&D. And my role principally is to evaluate sequencing and cellular analysis technologies, develop applications, and work with our faculty to ensure we have the technology, capability, and capacity to deliver our institute's scientific program. So I spent a very cold January day in our loading dock, watching our Ultima sequencer be unloaded off the back of a truck in January, several large crates. This is the sequencer, and after a lot of pushing and shoving, this is the sequencer, the Ultima UG100, and the AMP unit in our new lab, ready to go. So why Ultima? Well, we all know that the reduction in cost of sequencing over the past 20 years has been transformative. It's been transformative to our science, and it's enabled us to do a lot of things. But there are some experiments we're not doing today because sequencing is just still too expensive. And Ultima promises cheaper sequencing, which will enable us to sequence populations at greater breadth, i.e. larger PopGen projects, greater depth to see rare variation, and from the same individual at multiple time points during their life and during their disease. And we all know that over recent years, the traditional Moore's law graph has somewhat bottomed out, and a new technology is required to reboot this paradigm. So we signed up slightly before AGBT 22, but Ultima unveiled their technology at AGB 2022 and unveiled and said that at launch they will enable the $100 human genome. And this slide excited everybody because it infers that they can indeed do that, go even lower. So how do they do that? Well, essentially they achieve that through the use of an open flow cell, a spinning disk about 10 inches wide, and essentially they drop reagents on to a spinning disk. This creates a very thin film, enables them to use very little reagent, and so that's very cost effective. As opposed to an alumina type flow cell, which has a large void in the middle, and so a large amount of expensive material is wasted because only a small amount of the reagent required to fill the lane touches the surface, and also these flow cells need to be very precisely made and are very expensive. So it's this spinning disc that spins past a camera that enables it to be cheap through minimal use of reagents, but also by spinning past the camera, it's a lot more efficient in terms of imaging. So in Ultima technology, you do emulsion PCR, you have clonally amplified beads which get loaded into one of many billions of wells on this spinning disk, and they're sequenced as the disk is spinning. The instrument houses an auto changer, so I had one in the trunk of my car back in the early 2000s, and this goes back to that. Ultima features mostly natural sequencing by synthesis, which is a hybrid between traditional flow chemistry with unlabeled bases and sequencing by synthesis where you have reversible terminators. Here you have mostly natural, but a spike in of these fluorescent these reversible fluorescent bases, and they're present at a very low level. If you had all fluorescent bases, you would get quenching due to neighboring fluorescent molecules being next to each other and quenching the signal. So in order to maintain linearity and to reduce the cost, they have this mostly natural basis. So for any given template on a bead, a lot of the template copies won't get a fluorescent signal. Some will, and they will lead to a detection. And this detection is linear. So you get 3 bases in a row, 3 A's here. You get overall 3 times the signal because it's a flow chemistry. So 3 bases get incorporated during that flow of the A base. Here we've got 5 T's in a row. We flow T with the fluorescent base and 5 bases get incorporated, some of which are fluorescent, and we get roughly 5 times the average signal corresponding to 1. Now, we all know from these flow chemistries suffer from lack of linearity in homopolymers. Ultima seems quite good in that. They published several papers which show good linearity up to around 11 bases through homopolymers, which compares well to other chemistries like 454 and ion torrent. And they've done that, and they're improving on that by machine learning, proper algorithms that are able to look at a control molecule in the sequencing and revise the amplitude and the signal from each individual homopolymer length and correct for that for the real length. And this slide just shows that the qualities of the data back in June 22 at launch, and also at the time of this year's AGBT at February 2023, that shows that they've both increased their quality from Q30 of 85% to 89%. They've reduced their indel error rate. They've increased their read length slightly, and also increased their variant calling accuracy. So here we are. We have this largest sequencer, around 2 meters tall, at Sanger, that would allow us to do extremely cheap sequencing with short run times of around 16 hours. So what are we going to do with it? Well, for those of you who don't know, there are many available resources. So you yourselves can access some of the data from this website. You can get a reference human data set, and several manuscripts are available for common applications like whole genome synthesis, whole genome sequencing, single cell RNA sequencing, me-file-seq, and app notes for transcriptomics, me-file-seq, and single cell. At Sanger, we've just started sequencing our own samples. We're going to evaluate it across our programs for its utility in whole genome sequencing, low coverage whole genome, RNA-seq, single cell, me-file-seq, rare variant detection in cancer, microbiomes, and for high-C. And many of these applications so far have been demonstrated by other users. Whole genome sequencing, their ability to spot rare variants, in cancer, and also people have demonstrated concordance with Illumina data for single cell studies. Very briefly, for clinical variant detection, people have been able to detect all of the major variant classes. In tumours, they've been able to detect copy number variations, driver mutations, structural variants, and etc. High-C, the applicability of Ultima here is that you can get really deep data to show real high definition in terms of contact points in chromatin, to look at higher order regulation and higher order chromosomal contacts. In single cell, people have used it for both 5' and 3' with both 10x and parse to look at a variety of different experiments, and you generally get the same sort of UMAP cell types as you would for 10x genomics. People have demonstrated concordance with controls like the human reference RNAs and a good R² value compared to the same data with generated using Illumina. And likewise with me-fileseq as well. So it looks like virtually all the different application areas that people have looked at, you get very good concordance with other sequencing methods, apart from the fact that it's cheaper. So they haven't yet launched, but we wanted to engage at an early stage, basically to be involved and to help realize the promise of cheaper sequencing. So with that, I'd like to thank some other people at Sanger, Emily, Ian Johnson, our head of sequencing, Tom Preston, our procurement partner, Cordelia Langford, who's our director for scientific operations, as well as the many people at Ultima that we work with that have been a fantastic team and helped us to introduce us to this technology. Thank you very much.
Okay, hi everyone. My name is Delcy Martinez. I work at the Center for Advanced Technology at UCSF. And I just wanted to introduce you a little bit about us first. So the UCSF CAT is seen as the main NGS facility at UCSF. We're a hub for genomic work. So we house a lot of Illumina sequencers. So I'll be talking about the NovaSeq X, but we have 2 NovaSeq 6000s, 2 MiSeqs, an iSeq, and the HiSeq 4000. But we do have a lot of other instruments to help our users facilitate library prep. So we have a Beckman i5, i7, and an Echo 525, which is a tipless automated liquid handler. And on top of that, we are a sequencing corp, but we do a lot of R&D work on the NGS side. So we have collaborations with Beckman trying to miniaturize library prep, collaboration with CDPH as well, aiding in COVID surveillance, and other collaborations with our UCSF PIs working on single cell and spatial sequencing. But again, we are a sequencing course, so that's what we mostly do. And the idea is that the 6000 was the workhorse for a while. We were running about 1,200 S4 lanes since October 2020. So again, we were just seeing a lot of single cell libraries being submitted. So the idea is we actually only receive final pooled libraries. We QC them using ddPCR, and then we run them on the sequencer. So we just needed to increase our capacity. So we were quite excited that Illumina released the NovaSeq X. So the difference between the X and the Plus is that in the Plus you can run 2 flow cells. And I'm only going to focus on the 10B for this talk, but you can expect the 1.5 and the 25B to be available in the future. But for the 10B, it is an 8-lane flow cell. Again, the X Plus will take 2 of those flow cells. And for us, you can expect about 1.25 billion reads per lane. And they have 100, 200, and 300 cycle kits available with an extra 38 cycle for indexing. And then the workflow for the indexing is workflow B. So very similar to the 6000. All right, and so just some updates that are also coming along with the X Plus is, so we also had packing parties when we were dealing with the 6000. So the good thing is now reagents are actually shipping at ambient temperature, so it's a lot easier to deal with them. They're using lyophilized reagents, and just some simple tweaks actually help with our workflow. So the reagent cartridge actually has this clear thaw window, so you can visually inspect when it's ready to go on to the next step. And it's also going to allow for faster preparation of libraries for sequencing. You'll still need to prepare your fresh sodium hydroxide, dilute and denature your libraries, but now you're going to have to neutralize with this preload buffer that's provided. And you would load that onto a library strip tube, and instead of loading it into the flow cell like you would on the 6000, you'd pop that into your reagent cartridge along with the lyo insert, You could pop that into the instrument along with the buffer cartridge, and it should be that simple that you just hit start, and you'd be able to start sequencing. If you're doing paired N150 runs, run times we've seen less than 24 hours. So for us, it's a good, fast turnaround. And some additional updates have been done to the SBS chemistry. So the X-Leap SBS chemistry is supposed to be more robust, more efficient, faster, so forth, with the main difference being that the two-channel chemistry is green for T and blue for A. And we still need to test this with our own data in our facility, but it is expected to have less index hopping, and pre-phasing and phasing is also expected to be lower. And then in addition, you will have Dragon applications available directly on the instrument. So you could perform multiple secondary analysis in parallel. All right, so I'm going to preface this with, these are strictly UCSF internal rates. But the idea is we wanted to provide our users cheaper sequencing. So with the HiSeq 4000 being end of life, we needed an option to shift our users over. And so with a 10B 100 cycle would be a good option for those users. And we'd be able to keep that throughput in that we'd still have 8 lanes. But now our users are going to be seeing 3.5 times more data per lane. So each lane on the X is going to give you 1.25 billion instead of about 350 on the HiSeq, 350 million on the HiSeq. And we've managed to keep the price the same as what our users are seeing on the HiSeq. And the cost per million is less than $1 on the X versus close to 350 on the HiSeq. So again, just that cheaper sequencing. If we compare this to our users who are submitting libraries for paired N50, 10x attack, 10x RNA, sometimes we were getting those people on the SP flow cells. And again, 2 lanes comparison to 8 and more data, so 3 times more data. And it's actually going to be cheaper now for our users. So again, that cost per million is $1 on the X versus $450 on the 6000. All right, there are still going to be some instances where it might be more cost effective to stick to the 6000. And this could be instances where you have custom primers, custom run parameters, or just small scale runs. All right, and then again, we've seen an increase in single cell sequencing. So that's where the S4200 was coming in for the large scale projects. And the idea here is that you would be able to use the 10B 100 cycle option, and you'd be able to run 2 lanes to get the same amount of output as the S4, but it would be significantly cheaper, which is going to be great for the users. So again, that cost per million is less than $1 and close to $2 on the 6000, but it's going to be cheaper in the long run. And then lastly, for the users who are doing paired N150 runs, this is where the 10B 300 cycle option that we would offer would come into play. And again, running multiple lanes, and it would be still cheaper for the user than running a single S4 lane. All right, so hopefully it came across that ideally it's going to be every option on the X is going to be cheaper than what we have currently offered on the 6000. So cheaper and more throughput. All right, and now how did we go about implementing the X into our core? So on the right is the X Plus, and then we actually have our server rack right next to the instrument. It's a 10 gigabit connection between the instrument and our local storage. And then we have a 1 gigabit connection facing outward. And the reason for this is that we strictly only do run monitoring on base space. So we actually do not default to using Dragon currently. And for a couple reasons, being that we are core, we receive libraries from our users, so we don't, sometimes these users might not know what those barcodes are, what the orientation is. So demultiplexing is going to be a little bit difficult. So we need that extra flexibility. So we're actually still using BCL to FASTQ instead of the Dragon BCL convert. We don't upload sample sheets prior to starting a run as of right now. And as part of our workflow, we actually don't do any secondary analysis. We deliver the FASTQs, and then it's the user's responsibility to do the downstream analysis. One thing to note that is if Dragon were to be running, you wouldn't be able to start a new sequencing run. So that's something where, again, for a core, we just want to have fast turnaround time. And we've been working on loading concentration to help maximize our percent passing filters, because we've seen variation across different library types. And one of the ways we've been able to do this is by actually running a library and titrating it across an entire flow cell and seeing what the best match would be for that library. And also, one thing to note is that pre-run checks take 20 to 30 minutes. You're not going to get that nice little ping like you used to with the 6000. So we actually have to have someone currently there monitoring the system, making sure it's passing. And that's just something to keep in mind with your workflow, like how are you going to maximize your staff time and the efficiency of the instrument and its capabilities. All right, so we were, it's, pluses and minuses. We were the first NovaSeq X Plus on the West Coast, but with that, came the territory that we ran into a couple issues that maybe you guys won't. But one of the main ones that we actually had an IT requirement for a static IP that UCSF IT was actually changing our IP, and that was halting operations for actually a couple of weeks. We actually considered potentially being a dark site because we couldn't figure it out. And so a big shout out to Illumina for being very proactive to work with us and figure out the IT issue to either bypass it or get UCSF on board to figure it out. But for any actual instrument issues that we were experiencing, the idea is that future software updates are going to take care of those. One of the main ones to note is that we actually had to track our maintenance washes or when you'd have to replace an air filter. And that's not really something you want to think about. The instrument should be able to track that in the future. But I will say that of the runs we have completed, about 10, I would say, have met Illumina's specifications. So I'll just show you some data. So let's focus on lanes 7 and 8. So these are actually meeting spec. We're getting about 1.25 billion reads per lane. Q30 is good. But if we look at lanes 1 through 6, so these are now libraries that aren't doing so well. And when we try to figure out what the difference was, these are actually short insert libraries. So we actually think that the X might have a size bias, but we're still trying to confirm that that's the case. And again, this is where testing that loading concentration comes into play. And if you've ever had to debug a run, you might be familiar with this kind of plot, but it's percent occupied on the x-axis, percent passing filter on the y. And you'd expect your libraries to kind of cluster together as lane 7 and 8, which did relatively well, to cluster well on that percent passing filter and the percent occupied. But what we're actually seeing with lanes 1 through 6, which are short insert libraries, is that they're just kind of coming off of the diagonal. And what this is showing is that they're actually overloaded. So you can see that percent passing filter is actually dropping as your percent occupied increases. If the libraries had been underloaded, you'd expect that your percent passing filter would be decreasing as percent occupied decreased. So again, this is where future directions, just trying to figure out what the differences are between the libraries and how you can best plan for that with your loading concentrations and so forth. But with that, we're a very small team, but efficient. So I just want to thank my director, Chow, and then the three in the middle are the people who actually run the sequencers in our core. And again, thank you to Illumina for being very proactive and helping us with this new instrument and just install and everything. Thank you.
So before we get to the questions, can I get a show of hands on who's trying to get a sequencer right now or looking for the next 6 months? Not as many as I expected. Okay, questions? Bonnie.
Hi, I have a question about the singular conversion protocol. So you are doing 7 extra steps of PCR. Did you see extra singular PCR?
Yeah, so can I use this? Does it work?
Yeah, so my question was about the singular conversion protocol. So when you are adding extra 7 steps of PCR, are you seeing an increase in the PCR duplicate in the run? And how much you are seeing, if you are seeing?
I don't think this is working. 1, 2. Oh, that's great. Yeah, so that was one of the first thoughts, and I think we had a lot of questions from other people that reached out to us for early access to the instrument. We don't see anything. The results look very similar as if you ran it on an Illumina system. So no more PCR artifacts. We have Shosho here as well, who's running the instrument daily. I don't know if you have noticed any difference, but we haven't noticed anything on our fast QC runs regarding increase of duplicates.
So my question is kind of for the whole panel, and honestly, really the whole room. For those of you who've placed, and I know I've talked to some of y'all already, but for those of you who've placed an Element, an Ultima, a VD, a 2000, a NovaSeq X, give me the TLDR reason why you picked that instrument over the other ones out there. And if there's anyone who's seriously looking at an ONSO, I'd love to talk to you as well.
In our case, we looked at the NextSeq, the NextSeq 2000 and the Element Bio Aviti. There were no other options at that time when we were purchasing these ones. These were the choices, and the costs are just so dramatically lower on the Element Bio compared to the NextSeq 2000. Did not take much thought.
Yeah, I think for us, we do we do a lot of sequencing, so we just needed a high scale. We had a good relationship with Illumina. Tech support was also very big for us. So if our technicians did have, errors, it was very simple to get a quick, efficient answer from them. So that also played into our decision.
Yeah, for us, it was the ability to go from a very small instrument to a very large instrument on the same instrument. So you're going from a MySeq to a NovaSeq on your sequencer by keeping your sample cost stable. NextSeq 2000, unfortunately, was out of the discussion just because of cost alone. Between the other two systems, one was the flexibility, and the other was that it really fits very well with 10x runs, the multiplier of 2, 4, 6, 8 that I mentioned.
And my group is getting some new instruments too, and the main reason was we are running NextSeq 2000s, and it was the cost.
Okay, final question, because we need to give folks time to transition.
Hi, I'm curious about how each of your newer sequencers handle suboptimal libraries, whether that be less than ideal loading concentrations or not as diverse libraries, and how do you see that those handle them compared to normal libraries and compared to previous iterations of Illumina sequencers?
Well, I tried to address this a little bit in the talks. We have not done any extensive testing, any extensive stress testing. It looks like that if we don't have enough library, it's fairly linear, the number of reads that decrease the number of reads that you get out if you underload it. That's what it looks so far. It's definitely much more tolerant to low complexity stretches than the Illumina libraries sequences that we've had worked with. That's as much as I can say so far.
Yeah, same. We haven't done that many tests, but yeah, still trying to figure out the spike in concentration, see how that works out on the X versus the 6000.
In terms of testing, I agree we haven't feeded specifically very bad libraries to see how it goes, but when we see on the fragment analyzer a library we expect it to run well, that library runs well. When we see a library that is absolutely horrible and very, very low, I think similar, I would say, behavior if you run it on a NovaSeq. Potentially it's easier on the singular because you can load a single library on a single flow cell. So you don't have issues with titration, and it's very easy to get your library concentration right. And even if you do the F3 flow cell, you can still have one sample per lane or one sample in 2 lanes separately with the others. So it's very difficult to titrate it wrong. If your library is very bad, I don't think there is a sequencer out there that can save it. I wish there was.
So can I ask you about, you said you run amplicons on your machine. Are you, right, Giannis?
So what do you mean amplicons?
PCR products.
So, I mean, you have to, after you create your Illumina library, you have to pass it through PCR, correct?
Oh, that's what, okay.
So no, we only do 10x runs practically on the instrument, and we want to move to nanostring WTA runs as well.
In our case, the most stressing libraries are most like the TN-seq libraries. You're going from a low complexity stretch into full complexity, maximum complexity, and again, we ran them with 15% for X, and the data looked beautiful.
All right, we're going to wrap it up then. Thanks again for the speakers.